Next up, we have Dr. John Kulp, who is the CEO of BioLeap, Inc. He received his PhD in electrical science from MIT. Dr. Kulp has played a leading role in 7 successful business launches, including Symbolics, Inc. and Locus Pharmaceuticals. In 1985, he filed the registration for the first commercial.com domain name. Fun fact. Today, his moonshot is entitled Massively Scale Molecular Engineering for the War on Drug-Resistant Microbes. Hope you enjoy his presentation.
Thank you, Jackie. The emergence of drug-resistant microbes is viewed internationally as a major healthcare issue. If we don't cope with these superbugs and have treatments for them, this is going to go from a serious problem into a disaster. And that's why so many people are commenting on this. President Obama just recently this past fall issued an executive order to try to coordinate all the departments in the government around this issue and around this problem. But at the end of the day, we've got to come up with drugs that deal with it, and that's what I'm going to talk about. Where this concern comes from is that the incidence of these drug-resistant strains are individually becoming more prevalent among a given species, but in addition, more species are becoming drug-resistant. So you got this double whammy, and this is where people are really concerned. At the same time, the rate of introduction of new antibiotics is declining. So one hand, we got the problem getting worse. The other hand, the solutions are getting less. This is a cause for concern. The societal cost right now is rising, and it's really really a big problem that of the 2 million infections we get every year in the United States, half of them you get from hospitals, from going into the hospital. It's horrible, and people are dying. I have a little anecdote that one of my colleagues had a 21-year-old healthy daughter who went into the hospital to deliver a baby. She picked up one of these infections. They couldn't treat it. 10 days later, she died. And the baby was healthy, but it was a tragedy. So this isn't just hypothetical statistics. This is real pain. So there are many mechanisms for bacterial and microbial resistance, but at the end of the day, they all boil down to the fact that when the genes of the bugs mutate, that results in changes in the three-dimensional structure of the proteins that get expressed by those genes. And when those three-dimensional structures change, the ability of the drug to stick where it was doesn't stick there anymore because the structure changed. And so in this picture I have here, the gray surface there is the surface of a protein, and this is MRSA, a very familiar example. So the red gets changed to the green, and the drug no longer binds there. So the question is, how do we find drugs that bind to the proteins in ways that are insensitive to particular details? Over the last 10 years, my company BioLeap has been developing a molecular design methodology we call ligand engineering. The word ligand in this context refers to small little drug molecules that stick to big biomolecules such as proteins. That's what a ligand is. So the basic concept of ligand engineering is that you define a set of specifications in detail of how a drug molecule needs to bind in order to inhibit the function of the protein. Then you design custom molecules that implement those specifications. And the way we go about that is by coming up with maps of chemical building blocks and how those independently interact with the protein. And then we knit them together, link them together to make molecules. And then when we're done with that, we evaluate and prioritize them with comprehensive simulations. Unfortunately, my video doesn't work here. Video interoperability is not a problem that's been solved yet. So how do we apply this in the drug resistance context? Well, we would simulate fragment maps on a variety of different mutations, and then use that information to design the drugs. So what's the problem? I mean, this sounds great. It sounds like we can do it all. Well, the problem is it's too slow. This is basically a manual process. The ability to comprehensively search for solutions, we can only search for a few. And the second biggest problem is the fact that, well, this is great that this has been available to my company, but it's not available to the broader community, and it really needs to be if it's going to have more impact. Alternatives to this that have been traditionally used are what's called high-throughput screening of existing compounds. The problem is that the chemistry represented in that existing set of compounds has to be close to the final answer, or it's not very useful. And all the low-hanging fruit has basically been picked there, so it's, for these problems, it's not really working. Another method is getting fragment information, chemical fragment information from experiments, and that actually works pretty well. It's just very expensive. It's about 100 times more expensive than doing it computationally, so it doesn't scale. So our moonshot, if you imagine 1,000 infectious disease researchers that had 1,000 times more compute power applied to the ligand engineering, that would fantastically expand the ability to search and find these solutions. Now, where does this factor of 1,000 come from? Well, I have this idea called compute impulses, which is the ability to have a huge amount of computation for a very short amount of time. And this is what you can now do in the cloud that you could never do before. So it's at 100,000 cores or more for 2 days. And that buys us time. That's this really significant thing. But we haven't got the software to exploit this yet. And that's our fundamental problem. It's like I said, this is a software problem. It's an engineering challenge. It's not a scientific one. We don't need scientific breakthroughs to make progress here. But as I mentioned before, we need to make this available to thousands of researchers. We could use web technologies like OpenGL and WebCL and things like that to make molecular design software that just goes to any web browser. So the impact of this, again, nature is running ahead of us. I mean, its evolutionary process is extremely powerful. So how do we keep up? Well, we need to move faster, number one. Number two, we need to slow nature down. And the way we slow nature down is by finding ways to attack the proteins and whatnot that it's very difficult for nature to mutate around. Not everything is as easy to mutate around or get past as everything else. So we need to use tricks like that. If we can enable thousands of infectious disease researchers to have this kind of capability to much more effectively target and get compounds, thousands of lives can be saved, and the future threats that are coming down the road, we will have some tools to attack that. And I would mention, lastly, that this does apply more generally to other types of healthcare problems where mutations are at the core, like drug-resistant cancers and orphan diseases, subpopulation, personalized medicine, and things like that, all of which have in common the fact that specific protein mutations are key to the issue involved there. So thank you very much.
Thank you, Dr. Kulp. I wanted to open it up to the panel for some questions.
So I have a question. When the infectious disease mutates, is it changed in its entirety for that stream, or is it individual by individual? So is this problem going to be solved at the individual level or at the bug level?
It's got to be at the bug level because bugs communicate with each other and spread around. I mean, this thing about the incidence of mutated bugs, they spread from bug to bug to bug, and so it gets out of the individuals.
Right.
So that's why it's huge.
Great. Thank you so much for your presentation. My question has to do with the outcome versus the systemic factors that generate the superbugs, right? So you have the outcome, which is MRSA, and then you have the causes. So, for example, with regard to the hospitals, people not washing their hands, you have doctors overprescribing antibiotics to like an exponential degree, things like that. How would your technology sort of impact the structural factors that are causing the superbugs and the accompanying mutations?
That's a very good point. It doesn't directly, but I think the observation that one has to make is that those are all great things, and the government is trying to promote lots of programs, and hospitals are working very hard at this. The problem is compliance, and compliance not here but worldwide. And in fact, where this problem, I mean, we have this problem in the U.S. in a lot of ways, and because of hospitals, but it's 10 to 100 times worse in countries like, say, India, where the use of antibiotics is much less controlled, there's much less education about it, and these kind of issues of cleaning the hospitals and stuff are much worse. And then the bugs get from there everywhere around the world. So,
yeah, I guess just my question or my concern would be that this is a Band-Aid solution to some larger structural issues around.
Well, I think it's sort of the other way around. I think those compliance issues are Band-Aids that are not going to fundamentally solve the problem, and you've got to get down to, it comes down to compounds, or sometimes vaccines might work. There are some vaccines that are working in this. That maybe is another strategy. It's not probably as general as the compound strategy, but anyway, that's my thought.
Thank you.
So, I mean, typically the drug discovery process takes about 8 years from idea through clinical trials, and that requires a lot of money. Do you see this being supported through the pharma industry or through nonprofit agencies? How do you see this going from in this amazing idea into actually being implemented in the world today?
You know, that's an absolutely terrific question, is how do you fund this? And, you know, I mentioned that one of the reasons there aren't more antibiotics is an economic issue, that's why the pharma companies aren't investing in it. Number two is I submitted a grant to the NIH on sort of a smaller version of this same idea of basically making it available to more people and more researchers around. And what I got back on my score was excellent science, excellent impact, excellent competency to do the problem, and totally dinged on the business model that they just didn't believe. Well, maybe there are some business models, but we can think of. But that's the challenge. That's really the issue. I mean, it's like I said, there's a big engineering problem here with software, but funding it is the challenge.
Thank you very much.